My name is Dr. Salahuddin, working as Associate Professor in Noida Institute of Engineering and Technology, Pharmacy Institute, Greater Noida. Today, in this lecture, I will discuss about the medicinal chemistry involved in this anti-inflammatory agents. I have divided this topic into the two lectures. So, first lecture I will discuss some important class of the anti-inflammatory agents in this, and the rest one I will discuss in the next class. So, today this is the part 1 of the anti-inflammatory agents. So, first that we have to discuss about the anti-inflammatory drugs, the drugs which acts against the inflammation, and this agents that the agents act against the inflammations. There are two types of the drugs act against the inflammations. First one is the steroidal drugs, and the second one is non-steroidal drug. The basic difference between these two, as the name indicate, is steroidal drugs. So, steroidal drugs have the steroidal nucleus in their structure, and in the non-steroidal drugs that it does not contain any steroidal nucleus in their structure. So, the name that involves here, that is the non-steroidal. Today I will talk about this non-steroidal anti-inflammatory drugs, not the steroidal anti-inflammatory drugs. So, first here this non-steroidal anti-inflammatory drugs, that these drugs are used to relieve pain, reduce the inflammations. Inflammations means whatever the redness and the swelling in our body parts due to the some changes occurs in in our body. So, I will discuss in the coming slides, and it also used to bring down the temperature, the high temperature, that is called fever. This NSAID or nowadays are used to treat the wide range of the conditions. It is used in headache, it is used in painful periods, toothache, sprains, strains, infections such as the common cold or the flu inflammation of the joint, that is called arthritis, and some other tissues. So, here this non-steroidal anti-inflammatory drugs, how this non-steroidal anti-inflammatory drugs act? It acts by the blocking the production of prostaglandin. So, how this prostaglandin involved in the pain? In the pain and inflammation conditions, there is an increment of the prostaglandins and the leukotrienes. There are some these are the autocoids, and this prostaglandins, the chemical messenger, and this is responsible for pain, swelling, and some inflammations. So, we can use the narcotic analgesics and the non-narcotic analgesics. The analgesic that have the CNS effects, narcotic analgesic, non-narcotic, the analgesic that does not have the CNS effects. So, here this is the biosynthesis of the prostaglandins, prostacyclin, and the thromboxanes. Here the involvement of this there is two enzymes, this cyclooxygenase and the lipoxygenase, and when there is some tissue injury occurs it from the fatty acid, that is the arachidonic acid, the phospholipids, it is converted into the arachidonic acid. So, this arachidonic acid in the presence of the cyclooxygenase, in the presence of phospholipase from the fatty acid it got converted into the arachidonic acid, and this arachidonic acid it will converted into the prostaglandins. These prostaglandins are responsible for the pain and inflammations. So, our NSAIDs, the anti-inflammatory agents, blocks this cyclooxygenase, blocks the action of this cyclooxygenase. So, the overproduction of this prostaglandins is decreases. Now, here this is the complete the production or what is the effect of the COX-1 and COX-2, because the cyclooxygenase are of 3 types: COX-1, basically the COX-1 and the COX-2. In the books, the so many books, only the COX-1 and COX-2 are described, but recently the COX-3 also isolated, also discovered. So, here I will talk about this COX-1 and the COX-2. When the tissue injury occurs, it generates phospholipids, and from this phospholipids it will converted into the arachidonic acid, and this arachidonic acid converted into the prostaglandins in the presence of the cyclooxygenase. So, here there are 2 types of the cyclooxygenase. The COX-1, that is the constitutive enzyme that is present almost everywhere in our body, acts as housekeeping as a housekeeping effect of this COX-1, that is why it is a constitutional. And there is COX-2, inducible. It is inducible when the tissue injury occurs. So, both are involved, involved in the production of the prostaglandins. So, here our main focus with the help of this non-steroidal anti-inflammatory drugs to stop or to reduce the level of this prostaglandin and to reduce the pain and inflammations. So, there are 2 types of drugs that we can use mainly as an inhibitor, the selective COX inhibitors and the non-selective cyclooxygenase inhibitors. Selective that it will blocks the action of COX-1 or COX-2. Non-selective means it will act on both the enzyme like COX-1 and the COX-2. Now, the mechanism of action of non-steroidal anti-inflammatory drugs, as in the previous slide I have already discussed that there are 2 types of the enzymes are involved, that is the lipoxygenase and the cyclooxygenase. Here most of the most of the NSAIDs are the inhibitor of this cyclooxygenase activity, and when it blocks the action of this cyclooxygenase, then it will prevent the formation of the prostaglandin and reducing the sign and symptom of the inflammations, because this prostaglandin basically responsible for the inflammation. So, our aim is to reduce or to reduce the level of the prostaglandin in our body. So, with the help of cyclooxygenase enzyme, we can block this enzyme as an inhibitor, it can reduce these drugs by blocking the cyclooxygenase, it will reduce the level of prostaglandins. So, here there are two things, what is the basic difference between the NSAID and the COX-2 inhibitor. I have already told you that this COX-1 is the constitutive effect, and the COX-2 is inducible. When the tissue injury occurs, this COX-2 will produce, but this COX-1 is present in our body. So, this COX-1 is basically have the protective action, it acts as a protective action, and it will reduce the level of the gastric acid secretion by blocking this by the COX-1 enzyme. So, this the traditional anti-inflammatory drugs, aspirin, we are knowing about this aspirin. Inflammation is reduced by blocking the COX-2, but the protective action also of the stomach is also reduced because of COX-1 is also blocked. So, due to the blockage of this COX-1, due to the constitutive nature of this COX-1, it causes stomach upset and the bleeding from the intestine. And so, this selective COX-2 only blocks the COX-2 enzyme, that is the inducible enzyme, and it does not have any effect on the GIT. So, blocking this enzyme prevents the production of this prostaglandin. There is no effect on the gastric acid secretion. Okay. So, because of the selectivity of this COX-2, they reduces the peptic ulceration. This is the main feature of this COX-2. It includes the the inhibitors include, that is celecoxib, valdecoxib, rofecoxib and other members that we have. Now, to come to the important thing, important slide of the NSAIDs, the classification. The very important thing is the classification of NSAIDs. So, on the basis of their structures, the chemical structure of this non-steroidal anti-inflammatory drugs, that we can classify into salicylates, anthanilic acid derivatives, aryl acetic derivatives, the propionic acid derivatives, oxycam derivatives, pyrrolopyrrole derivatives, indole derivatives and the pyrazolone derivatives, all are the non-steroidal anti-inflammatory drugs. The drugs comes under these under the category of the different category, and the structure activity relationship I will discuss in the coming slides. Now, first the first category of the non-steroidal anti-inflammatory drugs, that is the salicylates. As the name salicylate indicate that it is a derivative of salicylic acid. Derivative of salicylic acid, and the two very interesting and the well known example of the salicylate, that is the aspirin and the sodium salicylate. They are these aspirin and the sodium salicylate, they are derived from the salicylic acid, and the salicylic acid we know that is the monohydroxy benzoic acid, and if the mechanism of action of this aspirin and the sodium salicylates inhibit the synthesis of prostaglandins and other mediators involved in the inflammations, and both have anti-inflammatory, antipyretic and analgesic properties. If I am talking about the structure activity relationship of salicylate. So, there are two, if I compare the activity with this structure, that is the structure of the aspirin. So, in this aspirin, that is the acetyl salicylic acid. So, here the substitution of the carboxyl group, if there is a substitution at this point, it may affect the potency and the toxicity. If we reduce the acidity of this carboxylic acid group by some other group with the less acidic, it retains the analgesic activity of the salicylic acid, but it is devoid of anti-inflammatory properties. Next thing is placing the phenolic hydroxyl group. Phenolic hydroxyl group, here this is the phenolic hydroxyl group, it is present at ortho. If we replace this position into meta or para, what will happen? It abolishes the activity, that means it always present at ortho position. Next is the substitution of the halogen atom at the aromatic ring, it enhances the potency. Substitution on the aromatic ring at the fifth position, what is the fifth position? 2, 3, 4, 5. So, here if we substitute by some functional group, it increases the anti-inflammatory activity. Now, come to the anthranilik acid derivative. So, first of all the structure of anthanilic acid, it is a N-aryl anthanilic acid. This is also called phenamides. These are the amine analog. There are some well known example of the anthanilic acid derivative, and it includes meclofenamic acid, flufenamic acid and the mefenamic acid. In the structure of compare this structure of anthanilic acid with meclofenamic acid, flufenamic acid and mefenamic acid, that there is only substitution at the N-aryl group. There is no substitution at this group. So, substitution at where? Substitution at second position, third position and sometimes at this position also. So, in the structure activity relationship of this anthanilic acid derivative, the carboxylic function, that is very important for the activity, whereas third and fourth aminobenzoic acid analogues are not active. Here the 2 aminobenzoic acid is present at the second position, third and fourth aminobenzoic acid, that is not, that will be not active. As we know that the replacement of this carboxylic group by the tetrazole. So, the carboxylic group, what is the relation between this carboxylic group and the tetrazole? This is the isosteric replacement, So, these carboxylic group and the tetrazole, these two are the isoester. And when we replace this carboxylic group with the tetrazole nucleus, that we can retain the anti-inflammatory activity. And the substitution on the anthranilik acid ring, that is generally reduces the activity, that is this ring must be unsubstituted. An aryl ring can lead to conflicting the result in the ultraviolet erythema assay of the anti-inflammatory activity. The order of the activity is generally 3 to 4 for the mono substitution, where the trifluoromethyl group, that this is present in the flufenamic acid, is the particularly more potent than the others. In the di-substituted derivatives, where the nature of the two substituents is the same, nature of the two substituent is same, like 2 chloro group, like 2 methyl group. So, 2,3 di substitution appears to be most effective, that is the 2 methyl group is present at second and third position in the mefenamic acid. The NH here, this NH of the antranilic acid is essential for the activity. So, the replacement of this NH by the other group, that it will produces the non-significant compound. Now, in the synthesis of mefenamic acid, it is synthesized from the 2-chlorobenzoic acid with this 2,3-dimethyl benzamine, and with the help of this copper bronze Ullmann condensation, it will produce this mefenamic acid. Now, the next category, that is the aryl acetic acid derivative. Aryl acetic acid derivative, that we can write the structure of the aryl acetic acid like CH2COOH. Phenyl acetic acid. So, we can say if R equals to aryl, that we can say aryl acetic acid RCH2OH. And the example includes of the aryl acetic acid derivatives, that is diclofenac, aceclofenac, felbinac and the bufexamac. These are the most important example of this aryl acetic derivatives. Diclofenac, that everybody knows about the name of this diclofenac, that the structure of this diclofenac, it is related with this structure. This is what? This is aryl acetic acid, and there is a 1 NH 2,6 dichlorophenyl attached at the NH group. So, diclofenac sodium is the effective member of this series and is well tolerated, and this is the inhibitor of this cyclooxygenase, that is COX-1 and COX-2. The aceclofenac, the another acetic acid derivative, that is this is the similar structure of the diclofenac. Not we can say the similar, that we have the CH2COOH, another the structure that we have again CH2CH2OH. Next is felbinac. The interesting thing is, in the structure of felbinac, it contains biphenyl group. So, in place of this phenyl, there is a 1 biphenyl group in the felbinac, biphenyl acetic acid. Next is bufexamac, is the phenyl acetohydroxamic acid derivative, and this bufexamac is used in the topical application. So, these are the some example of the aryl acetic acid derivatives which are used as the non-steroidal anti-inflammatory drugs. These are the some references. Thank you.